So chapter 31 is about plant responses to internal and external signals. I really think this is kind of a fun chapter. In a previous book, this was called plant behavior, which is kind of fun to think about because you don't really think about plants having behavior the same way that animals have behavior, but plants are still responding to signals, so that's technically behavior. So it's kind of a fun one, although it's not like you can train your plant to sit when you get the watering can out or something like that. But anyhow, we're gonna big picture talk about plant responses to signals, and then we're gonna talk about cell signaling and specifically different kinds of plant hormones, and then plant responses to environmental stimuli as well. So plants typically respond to environmental challenges by altering their growth patterns and their development, and most aspects of plant growth and development are under hormonal control. Again, this is a little bit different than animals. Like an animal can, run away from something if it's afraid of it. A plant can't really pick up and run away, so it has to grow differently. Tropism is turning of all or part of an organism in a particular direction in response to an external stimulus. In plants, this is usually a growth response. For example, phototropism involves both a cellular perception of light and a growth response of stem tissue to an internal chemical signal. And typically the plant's going to grow towards the light there. So to kind of remind you about cell signaling, hopefully you went over cell signaling in your Bio 160 class. If not, I believe it's in chapter 5. But cell signaling is a process in which cells perceive a signal and switch on an intercellular pathway that ultimately leads to response. And I don't know if two different people wrote the chapters or something like that in the book, but cell signaling as it's described in this chapter is slightly different than they describe it in the beginning of the book, which you went over in Bio 160, but it's really the same thing. You've got 3 stages. You have receptor activation, where a molecule in the plasma membrane or a molecule in the cytosol or the nucleus or the endomembrane system are going to receive a signal and become activated. These molecules are typically going to be a protein. And then you have transduction of the signal by way of second messengers. So second messengers can transmit the signal to an effector protein. So oftentimes calcium or cyclic AMP are second messengers. And then you have the cellular response, where the effector proteins are going to affect gene expression or other cellular processes that are going to influence the plant's behavior and development. So to kind of remind ourselves, let's label the steps involved in signal transmission. So response, reception, and transduction. So if we start here, we have a hormone or an environmental stimulus, and we have our receptor protein here. So this first step is going to be reception. And then we have our relay molecule, so our second messenger is going along here. So that's going to be signal transduction. And then you have your cellular response in this last step, which I probably should have changed this box here that says activation of cellular responses, because that should be pretty straightforward that it is cellular response. So when we talk about plant hormones are chemical signals that are transported within the plant body that bind to cellular receptors, thereby causing a response. So one hormone often has multiple effects, and hormones can be produced in very low concentration but can have a big impact on plant growth and development. And different concentrations or different combinations can produce different responses. There's 6 commonly recognized plant hormone groups. So there's auxin, cytokinins, gibberellins, Brassinosteroids, abscisic acid, and ethylene. And we're going to go over each of these in very kind of brief detail because we could really chew quite deeply into this, but we're going to kind of give it a good overview of all of these. And if you go to plant physiology class, you'll probably get more info on this. So if we start with auxin, auxin is often called the master plant hormone, and really there's several auxins. I pictured one for you there, but there's several of them because, again, these are groups of hormones. But generally speaking, auxins promote elongation in cells. They establish an apical basal polarity. What does that mean, apical basal polarity? So apex, tip, basal, base, so the up and down axis of the plant. It induces vascular differentiation, stimulates growth of the ovary, and reduces leaf abscission, and it mediates phototropism, so that response of bending towards the light. And auxin has many effects because it promotes expression of diverse genes that are known as auxin response genes. Under low auxin conditions, The auxin or IAA repressors prevent gene expression, but under higher auxin concentrations, this causes a breakdown of these repressors, and that allows for gene expression to occur. So when there's higher auxin levels, more genes are expressed, which means the plant can make more proteins. As far as auxin transport goes, auxin is produced in the apical shoot tips and young leaves, and it's directionally transported from one living cell to another. There's an uncharged form, IAAH, that can enter a cell by diffusion, but the charged form is an ion, and it requires a carrier, because remember, things cannot go across, like ions can't go across the lipid layer of the cell membrane. So an auxin influx carrier is needed to transport the charged IAA in the apical ends of the shoot cells, and then an auxin efflux carrier is going to transport auxin out of the cells. This happens at the basal end of the shoot cells. So if we look at this picture here, we've got our auxin charged form that can't just diffuse through the cell. You have an auxin influx carrier, then you have auxin efflux carriers here, and so we say there's polar transport of auxin or directional transport of auxin. So which way is auxin going to flow? If the influx carriers are on the top of the cell, the efflux carriers are on the bottom of the cell, so it goes in the top and out the bottom, in the top, out the bottom. So that means auxin is typically going to go down in shoots. You can see here that in this case, auxin could go to the side because the location of the transporters is going to really determine which way it goes. But typically, we expect auxin to go down in shoots. So auxin is important in cell elongation. We said that there's polar transport of auxin, and that's going to stimulate proton pumps or hydrogen ion pumps in the cell membrane. The proton pumps are going to increase the membrane potential and lower the pH in the cells. So this lowered pH is going to activate something called an expansin or expansins, which are enzymes that kind of loosen the cell wall's fabric, and that allows the cell to elongate. So here we have a proton pump, right? It's pumping hydrogen ions in, the acidity increases, that low pH activates expansins, that loosens the cell wall, and so that can allow the cell wall to expand, and also water tends to need to come in to kind of expand out that vacuole and push that cell wall longer or larger. As far as practical uses for auxin go, for fruit production, it stimulates flower ovaries to mature into fruits. Some home gardeners will spray something like a tomato with auxin to assure fruit production, but typically it's not profitable or useful on a commercial scale. Auxin is also important for development of roots on stem cuttings, and if you pinch the top of a shoot, you end up getting a bushy plant. If you remember, we talked about that in a previous chapter. We talked about releasing the axillary buds from apical dominance, but it does that because it disrupts that auxin flow from the shoot apex. So this is a really extreme example of pinching a plant to make a bushy plant, but you've probably seen that or maybe done that yourself before. And then also auxin slows premature fruit drop. So that brings us to cytokinins. Cytokinins increase cytokinesis. Great. What's cytokinesis? Cytokinesis is cell division, right? That's the final step. So you have mitosis, which divides the nucleus, and then cytokinesis takes you from one cell to two. So cytokinesis increases cell division. It's produced in actively growing tissues like roots and embryos and fruits. Makes sense. It can influence cell differentiation because they work together with auxin to control cell division and differentiation. It's involved in apical dominance, and it slows aging by inhibiting protein breakdown, stimulating RNA and protein synthesis, and mobilizing nutrients from surrounding tissues. So we talked in a previous chapter about having this certain ratio of auxin to cytokinin and being involved in tissue culture and getting either root development if you have a greater than 10-to-1 auxin to cytokinin concentration, or shoot development if you have less than 10-to-1 auxin to cytokinin concentration. That brings us to gibberellins or gibberellic acids, or sometimes just called GA. These are involved in stem elongation and leaf growth. They enhance cell elongation and cell division. They're produced in young roots and leaves and can induce bolting. So anybody who's a gardener, there's usually like one in every class, but hopefully it's you if you're watching this, right? So if you're a gardener and you're trying to grow like greens or something like cilantro, sometimes the plant will bolt, meaning you get these flowers here. So gibberellins are responsible for that bolting. In mini plants, both auxin and gibberellins must be present for fruit to develop. And if you've ever seen, you probably have, if you've gone to the store or gotten grapes, if you're like, man, grapes used to be smaller when I was a kid, maybe they were, because Thompson seedless grapes can actually be sprayed with gibberellins to increase their fruit size. As far as germination goes, after water is absorbed, remember we said imbibition is the first step in seed germination. So after water absorbed, release of gibberellins from the embryo can signal that seeds can germinate. That brings us to Brassinosteroids. These are chemically similar to cholesterol and the sex hormones of animals, which are also steroids, so that's why they're similar. Brassinosteroids can induce cell elongation. They induce the vacuole water, or the vacuole to intake water, and it influences enzymes that alter the cell wall carbohydrates. So you get cell elongation. They also induce stem division in the stem segments in seedlings, and they can slow leaf drop or leaf abscission, and they promote xylem differentiation. Brassinosteroids can also be applied to crops as a plant strengthening substance to help protect them from heat, cold, high salinity, and even herbicide injury. That brings us to abscisic acid, which slows plant growth and metabolism. Seed dormancy ensures that the seed will germinate only in optimal conditions, and in some seeds, dormancy is broken when abscisic acid is removed by heavy rain or inactivated by light or prolonged cold. And early germination can be caused by inactive or low levels of abscisic acid. Abscisic acid is also important for drought tolerance. It's the primary internal signal that enables the plant to withstand drought. And abscisic acid accumulation causes the stomata to rapidly close. Oh, there's some confusion though about abscisic acid. It's poorly named. It's named because we originally thought it was involved in abscission, which is dropping leaves and fruit, but actually that is auxin and ethylene that are more commonly involved in abscission. So abscisic acid has nothing to do with abscission. So sorry about that. That brings us to ethylene. Ethylene is response. Sorry, I got too excited. That brings us to ethylene. Ethylene is produced in response to stress, like drought or flooding or mechanical pressure or insect damage or infection. It's part of something called the triple response, which is going to slow stem elongation, thicken the stem, and increase horizontal growth. It's also part of senescence, which is the programmed death of cells or organs, and it's associated with apoptosis or apoptosis, which is that programmed cell death. It's important for leaf abscission, which is the leaf dropping, and I would say most famously ethylene is known for fruit ripening. So fruit producers can control ripening by picking green fruit and controlling their ethylene levels. So I don't know if you can see in this picture here, but that is like stacks and stacks of boxes of bananas. So bananas are actually picked green and ripened in facilities where ethylene is released into these special rooms. All right, let's review. Let's match the hormones and their responses. I think some of these are easier to do than others, so I'll probably start with the ones that I think are easiest first. And as always, if you want to read through everything on your own and hit pause and come back to check, you're welcome to do so. For me, the easiest hormone to match is ethylene, because ethylene is so important for fruit ripening. So I'm going to start with number 6, even though that's probably not the best place to start. But, you know, when you take a test, you do the same thing. You start where you can start. So ethylene is involved in fruit ripening. So 6 is going to be E. Another one that I think is easy to do is cytokinins, because cytokinesis, cell division. So let's see one that talks about cell division. So it looks like C, stimulate cell division, promote bud growth, slow organ death. Cytokinins, that's correct. So 2 is C. The next one I think of is Brassinosteroids, and I think like brass is strong, so that's your plant strengthening substance. So Brassinosteroids, B, plant strengthening substance, induce cell elongation and division, chemically similar to animal sex hormones. Chemically similar to animal sex hormones is another giveaway because steroids. So that is 4. So we did we did my 3 easiest ones there, so we're good there. Another one that I think is, I don't know, the next easiest for me to remember is abscisic acid, not involved in leaf drop or abscission, but involved in closure of the stomata. So let's see which one's talking about that. Oh, okay, up here, A, promotes stomatal closure, and Drought promotes seed dormancy. Abscisic acid, all of those match for that. So 5 is A. Then we have, I think we have gibberellins left still. So gibberellins are involved in like stem length or stem elongation. So gibberellin 3 is F, promotes stem elongation and helps seeds break dormancy and use stored reserves. And then I think the last one we have left is auxin. Auxin does a lot of things. Auxin's kind of that master plant hormone, but it's definitely involved in cell elongation, organ bending, like bending towards the light, and it regulates branching. Remember, if you pinch off the top of your shoot, your plant branches out there. So I think we got them all. So that's your matching hormones and the response of the plant to that hormone. As far as plant response to environmental stimuli go, since plants can't move, again, they have to adjust their development and growth patterns instead of moving away from things. The responses to light are photomorphogenesis. Responses to light include lack of light or darkness, the wavelength of the light or the color of the light, and the direction of the light and intensity of light. Plants also respond to gravity and touch and stresses. Stress for a plant is probably different than stress for you because the plant is not like, taking a test, studying for biology. A plant is not getting cut off in traffic, but a plant has to worry about drought and flooding. I mean, really, you have to worry about those things too, right? But plant has to deal with it more regularly than you do. Also, salt stress, heat stress, and cold stress. So those would be abiotic stressors because those are non-living, and then biotic stressors include herbivores and pathogens. So plants respond to darkness with something called etiolation. This is their morphological adaptations for growing in darkness. So again, if you've ever left a potato in a drawer and it has started to grow into a potato plant, you might notice that this potato looks like this. There's not a lot of green. It's just these white shoots that are going to be elongated. Or if you've tried to start plants on your own, like you tried to start seedlings for a garden, and you get these long, spindly things. Like, look at these pale, long, spindly plants. It's kind of sad. They don't have enough light available. So after exposure to light, shoots and roots can grow normally. However, if we're dealing with something like this seedling here, it just doesn't have enough energy. Like, this plant is not really going to be right again. But your potato, there's so much stored energy in the potato itself. The potato, after it gets into light, it responds normally. So etiolation is the growth adaptations for growing in darkness, and de-etiolation is what happens after the plant is exposed to light and allowed to grow normally. So if you remember, visible light is only a small portion of wavelengths available, and plants are going to only use certain wavelengths of visible light. An action spectrum depicts the relative response of a process to different wavelengths, and this is useful in studying processes that depend on light. And 2 major classes of light receptors found in plants are blue light photoreceptors and phytochromes that are going to absorb mostly red light. So if you maybe remember Bio 160, we talked about a little bit about photosynthetically active radiation or photosynthetically active wavelengths of light, and those are wavelengths of light that the plant can actually absorb and deal with. So if we look at our blue light photoreceptors, there's various blue light photoreceptors that control the phototropism, which is movement in response to light, stomatal opening, and hypocaudal elongation. And cryptochromes are going to help young seedlings determine if the light environment is bright enough for photosynthesis. If not, the seedling will continue to elongate. And this image, this is not from your text, this is actually from this paper here, but there's a mutant plant here that doesn't have the normal version of the HMR gene, and it makes them unable to respond to light, so they fail to produce chlorophyll, and they grow into the spindly albino seedlings that are going to die young. And here's a normal seedling in comparison there. Another kind of blue light receptor is phototropin, and this is the main blue light sensor in phototropism. So this phosphorylates when exposed to blue light, and it converts a light signal to a chemical signal. What's it mean when something phosphorylates? It means a phosphate group is added. So here's pictures, again, not from our textbook, but I think it's worth showing you pictures. In this experiment, they actually had a light source over on the side, and this is the wild type Arabidopsis seedlings, and this is a mutant that is deficient in this particular blue light receptor. So the wild type plant knows the light is actually coming from this way. The mutant version, no idea, growing up straight, up and down, because normally if you're a plant and you grow up straight, like that's where your light's going to be. Normally you're not a plant growing in a lab with a weird light source over here. So that brings us to phytochrome photoreceptors. These are red light receptors, and phytochrome can actually flip back and forth between two conformations. There's PFR, which is the conformation that only absorbs far red light and activates cellular responses, and then when left in the dark, the PFR transforms to red light absorbing PR. PR can only absorb red light and cannot activate cellular responses. So it's kind of like a light switch being in an on or off position, right? They're both the same thing, but there's different conformations there, and the only conformation the plant responds to that activates cellular responses is the FR version there. As far as phytochrome and germination goes, exposure to sunlight increases the ratio of PFR to PR in the plant, which triggers seed germination. So red light around 670 nanometers is going to increase germination compared with dark controls. Far red light at 730 nanometers is going to inhibit germination because the PFR is sensitive to the far red light and flips back to the inactive PR form. So the effects of red light and far red light are reversible, and the final light exposure is going to be what determines the response in the plant. So hopefully this slide kind of clears up the confusion about the phytochrome red and far red, or the PR and FR. So this is an experiment where seedlings were in appropriate conditions for germination, but they varied the light conditions. So in this petri dish, we have seedlings that were in darkness. We would not expect seedlings to germinate in darkness because phytochrome is going to remain in that inactive PR format. You know, there's maybe, like, it looks like this one doesn't follow the rules, but generally they're not germinating. This next set here, after a brief exposure to red light, that switches the phytochrome from PR to PFR, which is the active form, so these seedlings, or these seeds, are able to germinate and produce seedlings. Here, in this part of the experiment, they exposed it to R, which would say germinate, or red light, which would transfer the R to the FR form, which would say germinate. Then they turned on a far red light source, which would flip the FR to R, which would say don't germinate, and sure enough, most of the seeds did not germinate. Up here, they're like, let's just keep flipping switches, right? So we had exposure to red light, which would flip PR to the active FR form. Great, that would be germination. But then they had far red light, which would switch the PFR form to the PR form, which would say don't germinate. But then they flipped another switch and got red light again. So the PFR went to PR, and then the PR goes to PFR. So now we have the active form of the phytochrome, so the plant germinates. And that wasn't enough, so now we went crazy here, right? So here we have red light, which would flip the PR to the PFR form, but then far red light, which would flip the PFR to the PR form, which is inactive, then red light, which would flip the PR form to the PFR form, which would be active, and then far red light, which would flip the PFR form to the PR form, which is inactive. That was a mouthful, but ultimately you end up with the inactive PR form, so the seeds don't germinate, or most of the seeds don't germinate there. So phytochromes are also involved in shade avoidance. Phytochromes provide the plant information not just about whether light is available or not, but about the quality of light. Shaded plants receive more far red light than red light, so that means the PFR flips to the PR form there. So leaves in the canopy absorb the red light, so the phytochrome ratios are going to shift in favor of PR, and that stimulates the shade avoidance response. So this is going to induce vertical growth in the plant, because if you're a plant and you're shaded, you're not getting appropriate light, so you're just not going to do as well. Really, you can't get up and move. The only thing you can do is get taller. And so you can see here we've got the same kinds of plant in a high red to far red ratio or low red to far red ratio there. Again, red light is better for them than far red. So plants do have biological clocks and circadian rhythms. The circadian rhythm is the cycle that's about 24 hours long and governed by internal clocks. It may depend on synthesis of a protein regulated through a negative feedback loop. And both phytochromes and blue light receptors are involved in this, and phytochrome conversion marks sunrise and sunset, which provides the biological clock with environmental cues. So the photoperiod is the relative length of the day and night, and this is an environmental stimulus that's important for plants because they use this to detect the time of the year. Photoperiodism then is the physiological response to the photoperiod. And in the 1940s, researchers found out that plants actually care more about night length than day length, and flowering is controlled by the length of the night, not the length of the day. So I think probably you're aware that different plants flower at different times of the year, and we can classify plants as long day plants, short day plants, or day neutral plants. So a long day plant is a plant that flowers in the spring or early summer when the night period is short, because if the night is short, the day is long. A short day plant is a plant that flowers when the night length is long, because if the night is long, the day is short. So these are going to be plants that flower in late summer, fall, or winter. And then day neutral plants just don't really care. They'll flower regardless of the length of the night, as long as the day meets the minimum requirements necessary, to produce their carbohydrates to grow the plant appropriately. So here's just a couple pictures to show you the dark period time, daylight time, dark period time, daylight time, and the plant's response to that. So if we have a long day plant, it's going to flower when the day period is long. A short day plant would not. If the day period is short, the long day plant won't flower. If the day period is long, I think I said that wrong too. Sorry, this is a struggle lecture apparently for me, which is awful because it's one of my favorite topics. But okay, if the night is long, the day is short, a long day plant won't. We'll just pause it. We'll start over. So here's some pictures to kind of show you that. We've got this little graph here on the side, or this little pie chart here showing us the length of the dark period and the light period. And then we've got a column with a long day plant, an iris, and a short day plant, a goldenrod. And so, when the day is long, our long day plant is flowering and our short day plant is not. When the day is short, our long day plant is not flowering, but our short day plant is. However, if we experimentally break up the dark period with a flash of light in the middle of the night, our long day plant thinks that it is a long day because it's detecting a short dark period. Our short day plant thinks it is a long day because it's detecting a short dark period. So really the plants are determining the length of the day based on the length of the dark period, not the day. As far as like what kind of light matters for this night length in flowering, red light really seems to be the most effective in interrupting the night length for plants. So a flash of red light followed by a flash of far red light does not disrupt night length. However, some plants flower after only a single exposure to the required photoperiod, while others need successive days of the required photoperiod. And sometimes an additional environmental stimulus is required, like vernalization, which is a pre-treatment with a cold period to induce flowering. Is there a flowering hormone? Maybe. The photoperiod is detected by the leaves, which cue buds to develop as flowers, and florigen is a hypothesized hormone-like molecule that's responsible for controlling and or triggering flowering in both short day and long day plants. There's a potential gene locus that we've implicated for this, but we haven't been able to detect the hormone thus far in the plants. But we have evidence for this because if you graft plants together, they flower in response to kind of a systemic approach here. So here we have a short day plant and a long day plant, and we have a short day period for light. So our long day plant would not flower in response to this short day period or long dark period. However, if we graft the long day plant onto the short day plant, it does flower, which suggests that there's some sort of systemic response to flowering or cue for flowering. So that brings us to gravitropism, which is response to gravity. I don't know if you've ever put sod down. I did a little landscaping for a while in my youth, and they would always joke and say green side up, right? I still don't really joke, because if somebody pays to get their lawn sodded and you put it in upside down, I mean, they're going to be really unhappy. But plants on their own know which way is up and which way is down because they can sense gravity. Roots are said to be positively gravitrophic because they go towards gravity. Shoots are said to be negatively gravitrophic because they go against gravity. And plants may use statuses, which are these starch-heavy plastids, to detect gravity. However, some mutants lack statuses but are still capable of gravitropism, so that's probably not the only thing they're using. Maybe dense organelles or even just starch granules help contribute to gravity detection. Phytochrome-morphogenesis is a plant's response to mechanical stimuli. If you rub the stems of young plants a couple of times a day, it results in plants that are going to be shorter than control plants. Thigmotropism is directional growth in response to touch, and some plants have acute responses to mechanical stimuli and are called touch specialists. For example, the tendrils of a climbing plant here, they grow straight until they touch something and they coil around that thing that they've touched. Mimosa is the plant that has leaflets that, they'll be like this, and then in response to mechanical stimulation, they'll just collapse. And I'll post a video for you guys to see that if you're interested in it. But rapid leaf movements in response to touch result from transmission of electrical impulses called action potentials. And if you've taken anatomy or physiology, you've maybe heard of that. If not, we'll talk more about action potentials when we talk about muscle contraction and nervous impulses in animals. Plants also need to respond to drought. If they have excessive water loss due to transpiration, that can cause the plant to wilt and eventually die. Plants are going to try and reduce transpiration by closing their stomata. They'll roll their leaves to reduce the leaf surface area, which this tomato here is doing a great job illustrating that. They'll also drop their leaves as well. But all of those things have kind of a negative component that goes with them. If you close your stomata, you're not going to be able to do gas exchange for photosynthesis. If you reduce your leaf surface area, you're not going to be as effective at photosynthesis. And if you drop leaves off, you're definitely not as effective at photosynthesis. Plants can also struggle with flooding. I think this happens to people's house plants frequently. Like their plant looks bad because it's flooded, but people only know water. So they're like, oh no, I better water my plant. It looks bad. So these poor plants are suffocating in waterlogged soils. So roots need oxygen because roots need to do gas exchange and cell respiration. So roots do need oxygen because the final electron acceptor in the electron transport chain is oxygen. They can produce ethylene to kill root cortex cells, which can kind of create these air tubes or parenchyma that function like a snorkel for the plant to provide oxygen to those submerged roots. Salt stress is another big one. Salt can lower the water potential of the soil solution and reduce the plant's ability to take up water. So plants respond to salt stress by producing solutes that they can tolerate in high concentrations in their cells, which will draw the water into the cells. So that keeps the water potential of the cells more negative than the soil. Heat stress is another problem for plants. Excessive heat can denature enzymes in general, and excessive heat can denature a plant's enzymes. Transpiration, which is that water loss through the plant body surface, cools the leaves, but if it gets too hot, the plants are going to close the stomata to help reduce water loss, so that can only work so well. There are heat shock proteins, which can help to protect other proteins from denaturing during heat stress, and those are going to be produced at more high temperatures. So down here is Arabidopsis, mutants and wild type, and so there's one that overexpresses the heat shock protein, which is this OX here. The WT is wild type, and then the KO1 here does not express heat shock proteins. So if we look particularly at WT, OX, and KO1, the wild type, you know, we had heat shock occur. Wild type doesn't look too great, but it's okay. The overexpressing heat shock protein one is doing great after the heat shock. And then this knockout one without the heat shock protein is doing pretty terribly there. Cold stress is another thing. Cold temperatures decrease the cell membrane fluidity. However, the plants can alter their cell membrane lipid composition to maintain membrane fluidity. Freezing causes ice to form in a plant cell walls and intercellular spaces, and that's bad because water is going to leave the cells in response to freezing, which leads to toxic solute concentrations in the cytoplasm. However, frost-tolerant plants increase the concentration of non-toxic solutes inside their cells to reduce that water loss. And some plants have antifreeze proteins that prevent the formation of ice crystals. And the interesting thing about antifreeze proteins is it's actually evolved in many taxonomic groups, not just plants. And the 5 major classes of antifreeze proteins differ in amino acid sequence, so their primary structure differs, but their tertiary structure, so the structure of those proteins when they fold, is actually quite similar. So that's kind of interesting. Plants need to defend themselves against herbivores as well. Herbivory is when animals eat plants, and this is a big deal to plants, right? And if you think of like an animal eating you or eating chunks of you, that would be a big deal to you. So herbivory can inhibit growth, it can hinder resource acquisition, and it can increase the vulnerability of the plant to other pathogens like bacterial, fungal infection. Plants can counter excess herbivory a number of ways. They can have physical defenses like thorns or trichomes, which are those hairs. I think of like stinging nettle, right? That is definitely a good thing to keep me away. Chemical defenses like distasteful or toxic compounds, or behavioral defenses like recruitment of predatory animals. That one is the most interesting, I think, so we're going to kind of talk through that one. So here we've got this leaf, right? And there's a caterpillar that is wounding the plant. That's what it's doing. And there's the chemical in the saliva of the caterpillar. Both of these together start a signal transduction pathway, which causes the plant to synthesize and release volatile attractants. So these attractants go out into the air and they recruit parasitoid wasps that lay their eggs within the caterpillar. So the plant is like, ow, that hurts, that's awful, I'm going to call my friend and he's going to beat you up. And so the wasp comes in and lays eggs in this caterpillar, and if that's not enough, right, the eggs actually develop in the caterpillar and kill the caterpillar. So pretty interesting there, I think. Plants are able to defend themselves against pathogens to some degree. A pathogen, remember, is an infectious agent. So bacteria could be pathogenic, fungi could be pathogenic, protists could be pathogenic. So pathogens enter the plant through wounds or natural openings in the dermal tissue. So kind of the first line of defense in plants, just like us, is the outer layer. So their epidermis and periderm present a physical barrier which prevent pathogens from entering the plant. They also have an immune response. There's PAMP-triggered immunity. PAMP stands for pathogen-associated molecular patterns. And there's also effector-triggered immunity, which includes the hypersensitive response and systemic acquired resistance, which we're going to talk about all those on the next couple of slides. So the pathogen-associated molecular patterns, or PAMP-triggered immunity, is going to be when the plant recognizes particular patterns from pathogens. It's going to isolate and prevent the spread of the pathogen from the site of infection. And upon pathogen attack, the PAMPs are going to activate this pattern recognition receptor, which results in a signal cascade that leads to immunity. So here we have pathogen, and the cell is like, oh, I know you. And so it starts cell signaling, and you get an immune response in the plant. However, these things are kind of like an evolutionary arms race in a way. If you're a pathogen, that depends on infecting plants to survive. If you have some sort of advantage where you could avoid PAMP, then you would be able to infect the plant easier, and you would survive and reproduce more. So some pathogens have a way around this. So some pathogens release effectors, which are a pathogen-encoded protein that allows them to evade the PAMP-triggered immunity of the plant, and so that results in effector-triggered susceptibility of this plant. So pathogen, here it is. PAMP's like, oh, maybe I see you. Pathogen's like, no, you don't. Here's some effectors. You can't see me. And so that blocks the cellular signaling, and it blocks the immune response. However, like I said, it's kind of like an arms race. So if you're a plant and you could be pathogen resistant, you would survive and reproduce more. So if there's a mutation that allowed you to be pathogen resistant, then you would produce more plants like you. So in turn, plants have acquired resistance proteins that recognize pathogen-specific effectors. So that results in a secondary immune response called the effector-triggered immunity. So our proteins activate plant defenses by triggering signal transduction pathways, which include the hypersensitive response and systemic acquired resistance. So here we've got our pathogen, right? Here's PAMP. Pathogen's like, haha, effectors. But the plant is like, haha, our protein. So that allows for the cell signaling to happen and immune response to occur. So what's an immune response look like in plants? The hypersensitive response is an example of that. So the hypersensitive response is hypersensitive because it causes localized tissue death, as you can see in this tobacco leaf here. So if we can kill the cells and tissues in that particular area, that prevents the pathogen from reproducing in those cells, and that prevents the spread of that pathogen. This is particularly important if it's something like a virus that needs to take control of the host cells themselves to reproduce itself. And in the hypersensitive response, the plant produces enzymes and chemicals that damage the pathogen cell wall if it has it, interferes with pathogen's metabolism or pathogenic reproduction. So the systemic acquired resistance then is going to cause a plant-wide expression of defense genes and provides a long-lasting response to a diversity of pathogens. Methylsalicylic acid travels from an infection site to remote areas of the plant where it's converted to salicylic acid, which initiates the pathogen resistance. So here's our whole kit and caboodle here. We've got our pathogen, but it's intercepted here by the R protein. That causes the signal transduction pathway. We have a local hypersensitive response, but we also have a system-wide or plant-wide signal that goes out, and we get systemic acquired resistance. And so the whole rest of the plant acquires resistance to that pathogen. So let's match again we're going to match the stress and the responses here. As always, if you want to pause, feel free. So let's see. I think it's probably easier to read the big long response and then read the short word. So when would a plant adjust membrane fluidity, avoid osmotic water loss, and produce antifreeze proteins? Well, that last one gives it away. Antifreeze proteins, when's the only time it's going to freeze? If it's really cold. So A is 5, or cold is A. Formation of air tubes that help roots survive oxygen deprivation. That would be flooding. So 2 is B. Avoid osmotic water loss by producing solutes tolerated at high concentrations. That's going to be salt stress. So 3 is C. ABA production, reducing water loss by closing stomata. That's going to be drought. Drought and heat are a little tricky, but I wrote this so I know. But if you didn't write it, you could read E and answer based on that one as well. So E, so synthesis of heat shock proteins, which reduce protein denaturation at high temperature. So that's going to be a better match for 4 there. So we made it through 31. So if you need to go back to chapter 5 to review cell signaling, there's a little bit of it at the start of this chapter, but go back to 5 if you need to. We talked about the plant hormone functions. We talked about growing in darkness. We talked about photoreceptors and photoperiods and plants' response to night length. And we also talked about plant responses to environmental stimuli, both abiotic and biotic. As always, if you have questions, let me know.